this talk selected based on the submitted abstracts, and it is Chau, who is a PhD student in the Song Lee Lab at the Virginia Polytechnic Institutes. And she's going to be telling us about discovery of conserved marker genes for cross-species cell type assignment by machine learning. So super exciting topic. Can you share your slides? Perfect. Yeah.
Oh. Thank you, Kaisa, for the introduction. Can you hear me?
Yes, it's perfect. Take it away.
Yeah. Thank you, the organizer, for giving me a chance to present my project today. Good morning, everyone. My name is Chau. I am a PhD student at Virginia Tech. Today, I want to share with you about my project. It's discovery the conserved marker genes across multiple plant species by applying machine learning. In this project, I'm working with Dr. Timmy Sina and Dr. Song Lee, and Dr. Lee is also my PhD advisor. Well, in the past, we have bulk RNA-seq. It can only show the average gene expression across cells, but nowadays we have single-cell RNA-seq. It can show the gene expression by individual cells. So it provides a much deeper and more specific cell-to-cell variation. So we can investigate the transcriptome and identify different cell types for each individual species. So we can compare the single cell transcriptome among different species. From that, we can learn about the cell type evolutions, and we can transfer cell type knowledge across plant species. Well, we have two problems right here. First, in non-model plants, we don't know much about cell type specific marker genes. The second problem is distantly related organisms share very few one-to-one gene autolog. So comparative study between species is very difficult. Therefore, in this study, we propose a solution. We apply machine learning methods to select the relevant feature, reduce the complexity of the data, and improve the accuracy. In this study, we use the published data from 4 plant species. They are Arabidopsis, maize, rice, and tomato. First, we look through the root data of Arabidopsis, maize, and rice. Then we look for the conserved marker genes among them. Then we use the conserved marker gene to identify the cell type in a non-model plant, tomato. In this study, to identify the cell type marker genes, we applied 3 methods. They are rank sum test, sharp based on random forest, and support vector machine. For the rank sum test, we apply the Seurat package. It is a conventional statistical method. For the second one and the third one, we apply the XP marker, which is a Python package developed by our lab. There are also machine learning methods. You can see all the reference in the slide. Well, you can see right here, 3 plant species have different gene ID. Therefore, we have to apply AuthorFinder, which is a phylogenetic package. It allows us to convert the species marker gene to autologous marker gene. And in this study, we call them OMGs. Then we find the common OMGs. We came up with several results. The first one is comparison between 2 plant species. Totally, we have 3 heat maps for 3 comparisons. This slide shows one of them. This is a pairwise comparison between rice and Arabidopsis. We use the top OMG from 200 marker genes in each cell type cluster and find the common OMG between 2 species cell type cluster. You can see right here in the y-axis, is showing the cell type cluster of Arabidopsis, and the x-axis is showing the cell type cluster of Arabidopsis. We identify the cell type based on the list of marker gene from the reference paper, and or the number in this heat map is the number of common OMG between 2 clusters. Darker color is more common OMG, and lighter color is fewer common OMG. And you can see right here, all the red boxes are significant results were tested done by Fisher exact test. Well, the most interesting thing we learned from this result is that when you look closely into the green boxes, you can see that the xylem in rice share a large number of common OMG with the xylem in Arabidopsis. This also applies to cortex, the drygoblash or root hair, a drygoblash or nun hair, and phloem. However, it doesn't apply to endodermis. You can see the yellow box. There's only 6 common OMG between the endodermis in rice and the endodermis in Arabidopsis. However, again, you can see right here, the endodermis in rice share a lot of common OMG with a drygoblash and still in Arabidopsis. We'll highlight them in the red boxes. You can see it. Well, you can see right here. We're showing the structure of the root tip. It's showing that the steel in purple was very close to endodermis in pink. It suggests that these two cell types are functionally alike. However, the endodermis was far away from a drygoblash. So currently, we don't know exactly why these two cell types share a lot of common OMG. The second result we have is comparison among 3 plant species. Totally, we have 6 Venn diagrams, and this slide shows 2 of them. You can see right here. This is the cell type, and each circle is a plant species. And the number right here is the number of OMG, and in the center is showing the number of common OMG among 3 plant species. The most important thing we learned from this result is that xylem is the cell type that share the most number of common OMG among 3 plant species compared to all other cell types, such as root hair, nun hair, phloem, or cortex. The third result we have in this study is distribution of OMGs among 3 plant species. You can see right here, this is the cell type, and the y-axis is showing percentage of OMG. The x-axis is showing the plant species, and the color in the plot is the OMG components. You can see right here, the red and the orange box is showing share OMG with the same cell type. The next 2 colors is show share OMG with different cell type. And the last one is species-specific OMG. You know, the most interesting thing we learned from this result is that even though 3 plant species all look very different, there's only around 25 to 52% OMG is species specific. It means that more than 48 to 75% OMG is shared OMG in the form of either the same or different cell type. And then after we found the common OMG in 3 plant species, we use it to identify the cell type in a non-model plant. And the non-model plant in this study, tomato, you can see the 2 figures in the slide showing that after we have the list of conserved conserved OMG among 3 plant species, we can use it to identify the xylem and the drygoblash or root hair in tomato. The second discussion is comparison among 3 detection methods. Well, there are 2 things we experienced from the research method applied in this study. The first one is processing time, and the second one is the number of common OMG. You can see right here in the slide showing that the traditional statistical method, rank sum test, can provide the result fastest, and machine learning approach, specifically SHARP based on random forest, can detect the most number of common OMG among 3 plant species compared to 2 other methods. In this study, we make 4 contributions. First, this study allows us to detect the common OMG. Second, after we apply machine learning method, we realize that machine learning is very helpful to increase the number of common OMG among tree plant species. Third, after we found the common OMG in tree plants, we can use it to discover the cell type in non-model plants. And last one, this study allows us to determine the unusual common OMGs in monocot endodermis. Finally, I also want to appreciate everyone who supported me working on this project. Thanks all the members in my lab, our collaborator, the DOE funding, the GBCB program at Virginia Tech. Thank you for listening.
Thank you for the beautiful and exciting talk. I think we have a question from David Jackson. He would like to know, in your ortholog list, do you use one-to-one or one-to-many or many-to-many?
Oh, thank you for the question. You know that in this study, we expanded the orthology relationship. We not just focus on the one-to-one. We expanded to one-to-many, many-to-many, and many-to-one. Therefore, we can have more OMG orthologous marker gene, and then we have more common orthologous marker gene. So it's very helpful for the comparative study between species.
Excellent. Whilst we wait for further questions from the audience, I will ask a question of my own. So the unusual endodermis OMGs, do you think that that comes from the fact that maize and rice have also an additional barrier cell type, the exodermis? So can it be that some of the endodermal functions and typical marker genes are not anymore in the endodermis in these species?
Oh, thank you for the question. Actually, currently, we don't know exactly why the endodermis in monocots is share a lot of common OMG with the steel and the dragoflash. There are two reasons we thought it can be the problems causing it. First is they are functional divergent, and the second problem is that this can be mislabeled from the published data. So actually, that's the interesting question, and we want to investigate more in the second step in our research. Yes.
I think that it is the biology there that is interesting and not the detection method that you're using. So I think that the answers will be actually very interesting. It comes down to the evolution of the cell types. Yeah, that would be my guess. Okay, there's another question. So Med is asking, do you think you can use this model to identify OMGs in more forest plants or woody plants?
Yes, of course. We want to expand our research to more plants so that we can mapping the cell type in various species together. And further than that, we also want to apply more machine learning method, because we realize that machine learning can help to identify more highly specific marker gene compared to the traditional statistical method. So in the future, we will apply our study to identify the OMG in the woody or some other plant.
Yeah.
Thank you.
And then a question from an anonymous attendee asking if it's going to be possible to extend this method to look at family of proteins in non-model species based on domain structures. So I guess going beyond the ortho MCL, but looking at the domain structures as the...
Thank you for the question. Actually, currently, I don't know. Yes, I don't know for that question, but that's very interesting.
Yeah, maybe
and
food for thought.
We can learn it in the future.
Okay, then Ken Birnbaum asks, did the markers identified by the different methods agree on the specific genes? So you had
Yes.
the three, yeah.
Yes, actually, all three methods agree with the top highly specific OMG. So you can see, so you can see in my previous slide showing that all three methods found the different number of common OMG among three plant species, and among them there's some very highly specific OMG, and then all three methods will provide a consistent result. Yes, they all agree that the specific gene for three plant species.
Okay, questions keep coming in. Okay, so Vimal Kumar Balasubramanian asks, apart from orthology-based approach, what other datasets can be used to train ML model for precise OMG prediction? Yeah,
we use the published data. It is a including the gene expression data and the cell type. Actually, for this one, we use the list of marker gene from the published data to identify the cell type for to identify the cell type for all the species. And then we use that to train the machine learning model. So each cell type is corresponding to a class in machine learning model.
Yeah, I guess that the question was probing on the, like, if there's any other ways of doing it than giving it an like orthology already to get started. But so in your machine learning model goes the ortho MCL orthologs already. So is there a way of doing without having an orthology defined?
Actually for this one, we apply the machine learning method to identify the marker gene, and then we use the author finder to convert the marker gene into the orthologous marker gene. We don't apply machine learning model to identify the OMG gene.
All right.
So we apply machine learning to identify the marker gene.
Yeah. Okay. There's actually quite a few more questions in the Q&A. Thank you for those, but we are running low on time to answer all the questions. So maybe Tran, maybe you can type answers to some of the questions in the Q&A after this session or during this session, and we will move on to our next talk already. Thank you so much. Clearly a interesting talk with the many questions that are raised from the audience.